We're really excited to have you join us again. So next on our program is a session on selection that is chaired by Dr. David Israel. Dr. Israel is the Vice President of Lead Discovery Sciences at HitGen, where he oversees use and advancement of HitGen's DNA-Encoded Library platform. Welcome, David. Thank you for joining us, and thank you for all your efforts in organizing this workshop as well. The floor is yours.
Okay, yeah, thank you for the introduction, Serene. So we just heard two really nice talks on the design and synthesis of Dell libraries. And of course, the next step, once the libraries are in hand, are using the libraries in a process that's commonly referred to as affinity selection. So the next session of the workshop will focus on selections. And we have assembled three leading experts in the field to give talks on the topics of designing and making proteins for use in Dell selections, doing the actual Dell selection process, and then analyzing the output of those selections, largely with a focus on the molecular biology, the PCR and next-gen DNA sequence analysis. And those talks, respectively, are going to be given by Tracy Chen from HitGen, Svetlana Belyanskaya from Abgenix, and then Letian Kuai from WuXi. So the first talk of today's session is by Tracy Chen. Tracy is the Director of Biology and Assay and Screening at HitGen. HitGen, as a leading Dell CRO, has done many projects. Tracy has personally overseen and worked on greater than 100 Dell selection projects herself. Tracy is based in Chengdu, China, and due to the 12-hour time zone difference, has chosen to wisely record her talk. And we'll see that. Please do give the queue questions, and I will field the questions after Tracy's recorded talk on her behalf. So let me hand it over to Tracy now.
And hello, everyone. I'm Tracy Chen from HitGen, and my topic today is getting proteins ready for Dell selections. So this figure is a typical schematic flow of the Dell selection process. The Dell selection is an affinity-based selection by incubation of the target protein with the Dell molecules. After the incubation, we wash off the non-specific binders and then heat to release the molecules that enriched, and then repeat the selection by incubating the enriched molecules with the fresh proteins for another 1 or 2 rounds. After that, the enriched molecules are sequenced and the structures are revealed. We further synthesize the compounds of DNA and then validate those compounds in the downstream biochemical or biophysical assays. So for the whole part, I will mainly be focused on the target protein part, about how to prepare the protein before the Dell selection. So I will cover the four categories. It's about protein design, protein production, characterization, and pre-selection validation. So what makes Dell selection different from other high-throughput screening is that all the Dell molecules are attached to DNA tags. And also we utilize the immobilization step in the process to separate the Dell binders on the protein from the bulk Dell solutions. So this reflects in the protein design that we usually introduce tags to the protein. And also I will cover some other topics like different protein domains and different forms. For the protein production part, I will cover a brief introduction about different expression systems. For the characterization, this is the same for the other screens, just to ensure that the protein is of high quality. And for pre-selection validation, this is unique to the Dell selections, as we're including DNAs in the buffer, and we will immobilize the protein and validate that the protein is active under the selection conditions. So for protein design about the different tags, the tag can be introduced to either the N-terminal or C-terminal of the recombinant protein, but should be noted that the tag should not be buried inside of the protein or just interfere the interaction with the downstream binding partners or ligands. So in this case, if the C-terminal of the protein is involved in the ligand interaction, then the tag should be better be introduced at the N-terminal. This table lists the different tags that have been used. So for the first three, the His, biotin, and the Strep2 tag are preferred as they are with a very small size and high affinity to the matrix. For His tag, sometimes the non-specific binding is observed for the proteins without the His tags. And for biotin tag, the protein is usually expressed as the Abi tag in the construct. So just to make sure that the Abi tag is biotininated. For flag tag, the anti-flag beads can be used for immobilization. So as the matrix is antibody based, so usually we get a higher background in dose selection. For FC, GST, MBP, and SUMO tag, those can be used to increase the solubility of the protein, but it should be noted that because those tags are very large, ranging from 10 to 40 kilodaltons, so the tag unknown should be included in the selection to exclude the tag binders. And after we decided the tags, I'll talk about the protein construct. So for proteins with different domains, the domain of interest can be expressed instead of the full-length protein to minimize the effort in producing the protein. So for this case, the nuclear receptor here, there are two typical domains, the DNA binding domain and the ligand binding domain for nuclear receptors. So if the ligand binding domain is of interest, we can just express this domain unknown to exclude the DNA binding domain to minimize the effort in protein production and to exclude the possible interference introduced by the DNA binding domain in dose selection. And some proteins can only be expressed in a stable form with the adapter or scaffold proteins. The case here is the cerebrome. It's usually expressed with DDP1 as a complex. So the DDP1 unknown or the DDP1 complexed with other control proteins should be also expressed and included in the dose selection to exclude DDP1 binders. And some proteins, they bind the cofactors and form the holo form. So the conformation sometimes is different between the apo and the holo forms. So which specific form is of interest in the project and has to be decided, and the specific form can be prepared for protein design and production. The mutation of the active site is also a good strategy to differentiate the binding site of the DAO molecules and can be utilized in DAO selection. And for selectivity is another consideration for DAO selection, because a lot of proteins are very close in structure and active site. The case listed here is the D-ubiquitous. Also, the same goes for kinases. So including the counter targets from their close families can be a strategy to improve the selectivity of the DAO binders from DAO selection. So after we designed the construct, the expression, the protein can be expressed by either the prokaryotic or eukaryotic expression systems. So for both expression system, it includes the cloning of the targeted gene to the vector and introduce the vector to the host cells and expression and purify the target proteins. So for the two different expression systems, they differ in terms of speed, cost, and yield. So for the prokaryotic expression system unit, it has a higher speed, lower cost, and higher yield. But for proteins with post-translational modifications, unit, the eukaryotic expression system is utilized as it can provide the post-translational modification machinery to ensure the protein is expressed with good activity and the right folding. So for the protein QC and production is very important to ensure a successful screen, as we have compared commercial proteins with the same amino acid sequences, but they are from two different vendors, so they're expressed by different host cells. So for the proteins, they actually behave differently on the gel in terms of the fuzziness of the bind. And also for the selection, the result was quite different with some signals only enriched in one of the proteins. So a careful assessment of the protein QC and the production is recommended to ensure a successful screen. And so that's for the recombinant protein, but for some proteins are endogenous and extracted from biological samples. For those, they are without tags, and a biotinylation kit can be used to introduce the biotin tag to the protein. There are some commercial kits available, like showed in this case, the biotin group can be introduced to the amine on the protein. And after the biotinylation, we should validate that the activity is not compromised by either biochemical or biophysical assays. Then we can further test the immobilization of the protein by immobilize with the streptavidin beads. After we ensure that the protein can be successfully immobilized, so those tagless protein can be used in the DEL selection. Besides the biotinylation kit, some specific beads can also be used for immobilization of the protein without tags. I listed the two cases here are the cyanogen bromide activated beads and the carboxylic acid beads. Those beads have been used in DEL selection, and I will give an example about the carboxylic acid beads. So for these beads, it still utilize the amide coupling to immobilize the protein by the amine on the protein. And we have compared the performance of this immobilization method by using a recombinant protein that was expressed with his tags, and we immobilize the protein with either his tag or the or use this carboxylic acid beads, and they performed the selection, and the result was actually quite comparable for the two different immobilization methods. So this indicating that the carboxylic acid immobilization can be used to immobilize the protein without tags. And a hint here is that if the protein can be immobilized and tested still active in the SPR tested by amide coupling of the CN5 chip or the same related chip, then most likely the protein is active after the carboxylic acid beads immobilization. So for next, we will go to the protein characterization just to ensure that the protein is of good quality. Usually, we will confirm the molecular weight, the purity, and the multimeric status of the protein. I listed some example here. So for the SDS page is usually utilized to validate the molecular weight and the purity of the protein. Usually above 90% purity is preferred. And for ASAC, it's to validate the multimeric status. Usually like a protein with like monodispersity are preferred, but some like multi-dispersed samples have also been used in dose selection as long as the activity was demonstrated under the dose selection condition. And the mass spec can usually be utilized to assess the exact molecular weight of the target protein. And for some proteins, the multimeric status actually varies at different concentrations. So for those proteins, the stock concentration is another factor to be considered in the protein preparation. And for the storage buffer, usually, we just have to make sure the protein is happy in the storage buffer. Usually include a sodium chloride or a glycerol in the buffer to increase the stability. And as like imidazole or GSH or flag peptide are usually used for purification of the corresponding tagged proteins. So just to make sure those reagents are excluded in the storage buffer, as those will interfere with the immobilization in the downstream immobilization test. So, and also the activity confirmation is also necessary to validate the protein to ensure that the protein is suitable for use. I listed the different activity confirmation assays here, like the TM test to test the stability of the protein, or sometimes can be used to scan the buffers or confirm the two compound binding. Either the biophysical or biochemical assays can also be utilized to confirm the protein activity. And if a two compound is available for the target, it can also be used to validate that the compound is working as expected, either in the inhibition or like binding assay. So the assays listed here, but it should be specific to the target. So it should be most so the one chose there should be the most relevant ones. And after we validated the protein activities, we further tested the protein in some pre-selection validations to ensure the protein is active under the dose selection condition. So it involves some activity and immobilization test. So for a typical dose selection, it's and each round is around 2 hours at room temperature. So we usually test the activity up to 2 hours at room temperature, as this figure shows that there's no decrease of activity, indicating that the protein is stable. If we observe the decrease of activity, then we can adjust the selection buffer by addition of sodium chloride or glycerol in the buffer to increase the stability. And also incubation at lower temperatures like 16 or 4 degrees is another strategy. If all those didn't work, we can try to decrease the DAO incubation and immobilization time. So the purpose of this is to ensure that the protein is active during the handling. And then for immobilization, because we will utilize immobilization to separate the DAO binders from the bulk DAO solution. And so we usually perform the immobilization test to it's to like pin down the lowest bit amount for immobilization. as like higher amount of these will result in higher background in selection, and insufficient immobilization will result in the loss of DEL binders. So for the test, it's actually a bead titration test, which usually affects the amount of protein and then incubate with different volumes of beads and take the flow through after immobilization and wash it a little bit, and then take the beads, run the gel to determine the minimal volume of beads that can be used to immobilize the desired amount of protein. After we immobilize the protein, we further test the immobilized activity in selection buffer. So as it's DEL selection, and shared stem-sperm DNA is always included in the buffer as a blocking reagent to reduce the non-specific binding of the DNA tags. And sodium chloride and detergent are also included in the buffer to reduce non-specific binding, as we're dealing with urinary median or binion or trinium compounds. This is an example about immobilized activity test. So for the supernatant immobilization, actually there's no activity indicating that the protein is successfully immobilized. And we compare the immobilized activity with the free protein. Usually we observe a decrease of activity when compared with the free protein. This might result from the steric hindrance introduced by immobilization on beads. As long as the protein is active after immobilization, it should be compatible with selection. But we should rule out the interference introduced by beads during the assay. And if a two compound is available, we can also assess the two compound binding or inhibition under the selection conditions by comparing the, in this case, comparing the isophyty of the two compound on the free and immobilized protein. So in this case, the two compounds showed a good inhibition on the immobilized protein, indicating that the binding site of the two compound is accessible under the DEL selection conditions. So those are for the pre-selection validations before proceeding to DEL selection. But for some tough targets that they don't have a proper activity assay, so the on-DNA conjugates can be utilized for protein validation and selection condition optimization. So for on-DNA conjugate is to get a two compound and then attach the DEL DNA to this two compound. So to make this like a full-length conjugate as a positive DEL binder, and usually the conjugate without the two compound attached at the end can be used as a control to exclude the result from DNA binding. So for the conjugate test, we usually synthesize the conjugate and then use the qPCR to confirm the binding of the conjugate to the targeted protein. In this case, before making the conjugate, we usually have to make sure that the DNA attachment doesn't affect the activity or of this two compound. It can be analyzed from like SAR co-crystallization, or just simply tested after the DNA attachment by activity assays. And after the on-DNA conjugate can also be used as a positive control by spiking it in the DEL during the DEL selection. So this is a typical example of the conjugate validation. So for the DEL selection process, we usually immobilize the protein, incubate with the conjugate. This is as the input of the conjugate, and then wash a little bit, and then heat to denature the protein and release the binders from the immobilized protein. So this is the output of the conjugate. And we set the experiment like this. We usually incubate the full-length conjugate or the control conjugate with either the protein or the blank beads, and then compare the recovery of the conjugates in different groups. In this case, if the full-length conjugate showed enrichment on the target protein when compared with the blank, and also if the control conjugate didn't show any enrichment, in this case, indicating that the compound enriched under the dose selection condition. So the protein activity is ensured in this case. If we got like a similar recovery for the full-length conjugate and the control conjugate without any compound attached, this may indicate that the DNA binding dominated the recovery. And in another case, if there's no enrichment of the full-length conjugate or the control conjugate, so this indicates that there's no binding at all. Maybe the protein is not active under the selection condition, it has to be redesigned, or the selection condition has to be optimized. So that's for conjugate to validate the protein and the selection condition. And after all those are tested and validated, we can proceed to dose selections. But there are also some other frequently asked questions before dose selection. So I listed those here. One of the frequently asked questions is that is DNA binding protein compatible with dose selection? The answer is yes. And the DNA binding proteins usually can be divided into two categories: the ones that bind the generic DNA structures, or the ones that bind the specific DNA sequences. But both proteins can be utilized in dose selection. For the protein design part, we can prepare some mutants with the lower DNA binding affinity, or just exclude the DNA binding domain in the construct. But even we don't utilize those strategies, those proteins can still be screened by using other strategies during dose selection. And for RNA binding proteins, they can also be used in dose selection. Usually the RNA binding protein, it doesn't bind the DNA, and to ensure that we can start the selection, we usually test and confirm that the activity is not compromised in the presence of the of DNA, and the binding of the two compound is not affected. For some other specific protein, it requires some reducing reagent or metal ions in the buffer, so those can be included in the buffer. And some proteins, they have different conformations under different conditions. For some enzymes with a bi-substrate, they they actually show the different conformations with different substrate or cofactor present. So those have to be taken into consideration when preparing the protein and design the construct. For the protein amount, usually a typical dose screen would require 100 micrograms to several milligrams of the protein. It depends on the size of the protein and the selection design. The dose selection process is a binding process that is driven by the protein concentration. Usually the higher protein concentration would result in higher signals, but 1 to 5 micromolar of the protein is the concentration usually used in dose selection. And we usually use immobilization in the dose selection process. So there are two different selection mode: in solution mode or immobilization mode. They differ in the order of DAO incubation and the protein immobilization step, but in most cases, the two selection mode, the performance are quite comparable. So that's all for my talk. To sum up, I have covered the protein design, protein production, characterization, and pre-selection validation in this talk. And some take-home messages here is that we have, as we're going to immobilize the protein, so make sure the proteins are with the tags, and also we will immobilize the protein and make sure that the immobilized protein is active, the protein under the DEL selection conditions. And like any other traditional screens, the protein of high quantity is the prerequisite of the successful screen. So always make sure the protein are with a good purity, activity, with the right multimeric status and confirmation. So that's all for my talk today. Thank you guys for listening.
Okay, well, thank you very much, Tracy. Again, that was recorded. It's 4 a.m. in the morning in Chengdu. So I hope, Tracy, you're asleep and not watching this live. You said you might tune in, but you can see this recording afterwards. I will take some of the questions on Tracy's behalf, as I mentioned. So I'll just start at the top and go work my way down. Tracy, could you please comment on the importance of appropriate buffer systems, cofactors, et cetera, for efficient DEL selections? So yeah, having the right buffer where the protein is well-behaved, happy, fully active, et cetera, is critically important to the process. And Tracy did just go through some of the pre-selection analyses that's done to address the activity of the protein, and that is often done in different buffers, including starting with the buffer that's known to be optimal for the protein, and then sort of working our way towards the DEL selection buffer. Cofactors are often included. Again, it's case by case, and it's based on the known biochemistry of the protein. But it's often included on, in a plus-minus situation in a selection where one sample might be done in the absence of a cofactor, and a parallel sample may be selected in the presence of the cofactor. And that is actually often very informative on the binding site of different hits. And in some cases, what we do see are hits or binders that are dependent on the presence of the cofactor. They don't necessarily compete at the same binding site. Next question from Jose Santos. Does screening of a non-monodisperse or aggregated protein lead to the identification of false positive binders to the monodisperse protein? Well, certainly screening of an aggregated form of a protein can lead to artifactual false positives, and we try to avoid that at all costs. If it's a mixture of monodisperse and either multimeric or aggregated, there still may be authentic binders that come through the screen, but it's often difficult to distinguish binders to the monomeric protein from an aggregated form. Sometimes tool compounds are useful, again, in a plus-minus comparison to identified binders that are to sort of the authentic form of the protein. Next question, do you have comments on tricks or concerns when attempting a DEL selection with multimeric target that is not fully one species in the screen? This is from Nick Simmons from Jensen. Yeah, you know, if it's a multimeric protein that naturally occurs in different states, different multimeric states, I don't think that's a big concern because, you know, ideally, you know, if proteins are dynamic, and that's the normal physiology that the DEL selection would mimic the normal physiological condition. So if there are multiple multimeric states that naturally occur, I think that's okay in the DEL selection. Again, I don't know if I call this a trick or not, but including either tool compounds, or we heard briefly from Tracy on the use of DNA conjugates as sort of positive control compounds that could be spiked in. Those often give an element of confidence that the selection is working and that known binders can be pulled out, when they're made as compounds that mimic compounds in the DEL libraries. All right, so next question, I'm just going down the list here from Alex Hurd. Does anyone do SEC instead of immobilizing proteins? So I assume that means in the actual selection process and not for purification. So SEC would be a little difficult. The DNA itself is on the DEL library is quite large, over 100 kilodaltons. So the DNA protein complex obviously is larger than just the protein alone, but a lot of that will be contributed by the DNA. So it may be difficult to separate protein DEL complex from free DEL using SEC. There are sort of tagless or beadless, I should say, rather beadless selection schemes that have been described, mainly using capillary electrophoresis to try to separate free DEL from DEL protein complex using CE as a method, not SEC, but it's, I guess, conceptually similar. Okay, so another question from Jansen, Rodrigo, this is from Rodrigo from Jansen. For a protein with no tag available, which is best, non-specific biotinylation or carboxylic acid bead capture or other? That's a tough question. I'd say it's case by case. The vast majority of the targets that we work with are tagged, using sort of a recombinant HIS, and one of the biotin tags are the most common. In cases where there is no tag available, my preference, I'm always a little hesitant to do any chemistry on a protein, but my personal preference might be a chemical biotinylation, just because then you still have the power of streptavidin biotin to work with, and it might be slightly easier to characterize than capturing a protein on the bead, as far as the QC of the protein after the capture. One other strategy, which I guess would be in the other category, would be if there's a natural or a known binding partner, either a peptide or another protein, or something like heparin, for example, that can be used to capture the protein. Even though the protein is not tagged, you might be able to use a known ligand, again, whether it's a natural interacting protein, peptide, or compounds such as heparin, to do the protein capture and use that in a selection. All right, we have a couple more minutes and then we'll move to the next talk. Do you typically screen monodispersity and stability in the DEL selection buffer? So we definitely screen for monodispersity, usually not in the DEL selection buffer, because the final selection buffer contains the DNA from both the sheared salmon sperm and the DEL library itself, and that makes some of those assays difficult, I think. But we do definitely screen for monodispersity in the assay buffers, or the buffer that the protein is provided in when possible. The stability, as Tracy outlined in her talk, is done under DEL selection conditions, to mimic the conditions that the protein will experience in the selection. So stability, activity stability is definitely done in DEL selection buffer. Can you comment on membrane proteins in DEL selections? Yeah, that's one of my pet projects. There's a poster on the wall back there showing just that. You can do membrane targets. The real challenge is expressing enough of the protein to have it in high enough concentration that it drives the affinity binding process. And for some proteins, it's a little bit more straightforward to do. You know, we published using GPCRs in membranes and whole cells for selections. Other proteins are much more challenging to achieve the levels of expression required to drive that affinity process. And I think I'll end there. I'll try to get to the other ones by typing, and then move to Svetlana's talk. And so again, Tracy, thank you for that talk.